Today we'll discuss about internal structure of spleen and review histology of spleen. As understanding of internal structure of spleen helps in interpretation of histology of spleen. To understand internal structure of spleen, we need to understand concept of white pulp and red pulp. Cut surface of spleen on gross examination appears to have some areas that appear white and that's called white pulp, and other areas appear red that are called red pulp. To better understand internal structure of spleen, we need to understand functions of spleen. One function of spleen is to remove old and damaged red blood cells from circulation. And spleen is site for adaptive immune response for blood-borne antigens and pathogens. Internal structure of spleen is based on circulation of spleen. Splenic artery divides into trabecular arteries, and trabecular arteries then divide into central arteries. Around the central arteries, T cells are present in form of a rim, and this is called periarterial lymphatic sheath or periarteriolar lymphoid sheaths. So this is T cell zone, and T cells are present around central arteries. Adjacent to T cell zone, B cell follicles are present, and these follicles are supplied by branches of central artery. And like a lymph node, after interaction with antigens, these follicles develop pale appearing germinal centers. So this part of spleen containing B and T cell zones around central artery and its branches is called white pulp. And under microscope, this area appears to have purple cells on white background. And purple cells are due to color of nuclei of lymphocytes. After central arteries leave white pulp, they divide into branches and ultimately form capillaries. These capillaries are surrounded by rim of macrophages, like central arteries are surrounded by rim of T lymphocytes. So these macrophages phagocytose any damaged or old RBC that leaves these capillaries. Other characteristic of these capillaries that make them unique is that these are open-ended capillaries. Capillaries normally join postcapillary venules and this form a continuous circulation. However, these capillaries end abruptly in substance of spleen. This results in pouring of blood containing blood cells and any antigen and bacteria present in blood into substance of spleen. So this is red pulp of spleen. It appears red due to presence of numerous RBCs that have left circulation through open-ended capillaries. Red pulp of spleen has two important structural components. One is venous sinuses and second is splenic cords. Venous sinuses have gaps between adjacent endothelial cells and they also have discontinuous basement membrane. The gaps between endothelial cells are small, however, red blood cells can pass through these gaps and re-enter circulation. Splenic cords have network of connective tissue, blood cells, and any pathogens or antigens that were present in blood. So how red blood cells in splenic cords re-enter circulation? To re-enter circulation, red blood cells have to pass through connective tissue network of splenic cords and through small gaps between endothelial cells of venous sinuses. So only red blood cells that have flexible membranes and can deform enough pass through splenic cords and then through walls of venous sinuses to reach circulation. Old and damaged red blood cells that cannot deform and cannot pass through these channels are then phagocytosed by macrophages present in splenic cords. and any bacteria present in splenic cards are also phagocytosed by macrophages. Another feature of B-cell follicles in humans is that capillaries at margin of B-cell follicles are open-ended capillaries, and these capillaries allow passage of bacteria and also passage of lymphocytes from circulation to this part of white pulp. B-cell follicles are surrounded by perifollicular zone or marginal zone. This zone contains macrophages. These macrophages pick up any bacteria, and these macrophages are also antigen presenting cells, and they present antigens to lymphocytes. So internal structure of spleen consists of white pulp and red pulp. White pulp consists of T cell zone and B cell follicles. Depending on nature of antigen, immune response can be antibody mediated or cell mediated response. Under microscope, white pulp appears to have purple cells on white background. Red pulp appears red due to presence of numerous red blood cells. And red pulp consists of venous sinuses and splenic cords. Now let's see this in a histology slide. White pulp appears to have purple cells on white background. So this is white pulp. This is white pulp. This is white pulp. And here we can see a central artery that is surrounded by periarteriolar lymphoid sheath. In contrast, red pulp appears more red. This is red pulp. This is red pulp. This is red pulp. So red pulp appears more red. On higher magnification, we can identify venous sinuses and splenic cords. So these are venous sinuses, and these venous sinuses have cells inside, and intervening are splenic cords. So we discussed that spleen consists of white pulp and red pulp. White pulp consists of periarteriolar lymphoid sheaths and lymphoid follicles that contain B cells, and red pulp contain venous sinuses and venous cords. In splenic circulation, central artery is surrounded by white pulp, then it supplies red pulp and ends in open-ended capillaries. Blood is then collected in venous sinuses present in splenic cords, and these venous sinuses then join veins.